I want to welcome you to today's Key Opinion Leader call with Dr. Jim Lalonde. I'm Lou Basenese, MDB Capital's President and Chief Market Strategist. Jim, welcome to the call. Appreciate you making time today.
Yeah, thank you. Happy to be here.
Yeah, well, I want to cover a couple housekeeping items and then brag about you a little bit because it's always easier for someone else to do that. So I want to let everyone know first and foremost, today's call is going to be recorded. I just wanted to make you aware of that. We are also going to have a brief question and answer session following the call. So if you have questions that come up along the way, I just encourage you to use the Q&A function in Zoom to submit those questions so that we can see them as they come in. It'll just facilitate a much more efficient and smooth Q&A session as we get to the end. The other thing I do have to mention, because we will be discussing a company that has filed a S-1 registration statement for an initial public offering that is not yet effective, but has nonetheless been filed, and MDB Capital and the broker-dealer are acting as the sole underwriter, we have to provide some forward-looking statement disclosures. There is the possibility that we will make some forward-looking statements during today's call. We ask that you refer to the latest S-1 filing amendment to understand the risks and uncertainties that can come with any potential investment. And we also just disclose up front that this is not an offer to buy or sell or solicitation to purchase anything. So we just want you to be clear on that. And with that being said, I'm going to move forward and more properly introduce you to our key opinion leader here today, Dr. Jim Lalonde. He is someone that is very well known in the synthetic biology space. I will tell you that he has more degrees than I can count and much more experience. And what impresses me the most about it is it's across a broad range of science and disciplines, as well as commercial expertise. So I'm excited today to get into some of that background, but then also Invizyne Technologies and what excites him about that technology and the potential opportunities. So Jim, welcome again, and thank you for joining us.
Yeah, no, thanks for having me. Excited to participate.
So I'm going to start with the end in mind because I'm an investor first and not a science person, although I'm a layman in that regard. So let's start with, given your career, what excited you so much or enough to bring you to the point to join Invizyne's board? And I'll preface that with, you're also involved with several other startups as scientific advisor and board member. So just curious in how this fits into what you're seeing in the science world and what excites you about it.
Yeah, no, I've always gravitated towards science producing products of value to society, and excited by enzyme engineering and biological strain engineering to produce materials. And most of my career has been in that area, and I've just been privileged to be part of the whole community and see things transition from kind of a boutique-y, small, niche science to now we're talking about impacting, global energy supply, for instance. And so it's really, really cool. So that's kind of what motivates me, is doing great science, but with the end in mind being a product that people are going to use and it's going to change society. So when I saw Invizyne and taking what nature has done and putting it in a format that's more amenable to things we want to manufacture in the mode that we want to make it, instead of growing things or feeding animals and then harvesting, putting things in a more manufacturing format so that we can make materials at low cost and really help the sustainability of this planet.
Yeah, no, that's a great insight there. I just wanted to just kind of, let's define some terms to make sure everyone understands. When we talk about enzyme engineering, how would you describe that to the layman? I mean, give them the quick science on what's an enzyme's function, what does it do, and then why does that need to be engineered for what we're talking about here?
Yeah, so enzymes are the catalysts that really drive all life on Earth, right? And they're proteins that do all the chemistry that we need to survive. They take, sunlight hitting a plant and converting that into energy like sugars and fixing CO2 from the atmosphere. That's done with enzymes. The way that we live, and we eat food and we have thousands of enzymes in our body that convert it into tissue and into even the way we store our thoughts and the energy that we use to exist. It's all mediated by enzyme catalysts, and nature's done a fantastic created a fantastic algorithm for for adapting enzymes to a changing environment. And so what we've been able to do is harness that for our purposes to adapt enzymes to make stuff that we want to make. For instance, jet fuel, at a low cost. So really enzymes are the catalysts that drive all life on Earth, and energy conversion of that, and creation of matter, and enzyme engineering then is taking those enzymes and modifying them for our purposes. So the Nobel Prize in Chemistry was awarded in 2018 to Francis Arnold for directed evolution, and that's kind of the type of technology that I've used in my career, where nature creates new enzymes by having random mutations and changes, and then if the environment has changed, like say this, for instance, the CO2 in the atmosphere is going up, then changing an enzyme, the way it functions with these random changes in nature, these mutations, allows that organism then to adapt to a new environment. And so we can do that same thing in the lab, but we can speed it up. Instead of taking thousands or millions of years, we can do it in days and weeks by creating libraries of these enzymes and then testing them for the new function that we want to do. For instance, making isobutanol as jet fuel.
Right, which I believe is one of Invizyne's target markets eventually, potentially, that they've talked about. So help me understand too, because you've been involved in this space. I think it was your postdoc studies in 87, 89 in biocatalysis. So how have things changed from that time when you're talking about enzyme engineering to advance? What are the big advancements that happen to now? Because you're talking about being able to engineer in days and weeks versus months, years, and longer than that.
Yeah, no, when I started out in the 80s, I was a straight organic chemist using old-fashioned, still obviously valuable technology, using organic chemistry to make stuff. But kind of a lot of it is brute force, things happening at high temperatures and making not just the thing you want, but other things. And so when I saw what, at the time, Chi-Wei Wang was a professor at Texas A&M using enzymes to make stuff, I was really excited to see the selectivity and the precision that these work at. So rather than brute force, it was a very precise tool. Even that being said, though, they're still like Stone Age compared to where we are now. It was like working with rocks and sticks. We could take enzymes from nature, and we could try to make them do what we wanted. And there was a few enzymes that would do that, but the vast majority of them were very precise for doing things in a body or in a bacteria, and they didn't work outside that environment. So we had no way of engineering them in those days. And so we just took the few enzymes, and everybody published papers on one enzyme, and there was hundreds of people doing the same thing, basically. So when, directed evolution came along, Francis Arnold and then Maxigen and Codexis, that gave us the ability to make these custom enzymes bespoke for whatever we wanted to do. So that opened up a whole array of chemistry that we could do, not just that one enzyme that happened to work, but really thousands of enzymes that we can adapt and create, quite rapidly in the lab. So.
Yeah. So with that in mind, now that we have these tools and Invizyne is capitalizing on that, is there a limited universe of enzymes that Invizyne's technology could apply to from your perspective? Is it cover the majority of the market? How does that flesh out, given your experience and knowledge with the whole side of the science side of the business?
Yeah, no, there's really little limitations on it, and the kind of the whole area is blowing up. That now, we have high throughput ways of discovering new enzymes, and people are adding literally 1 billion genes that code for enzymes to their databases every year. So there's really no limitation. What we're doing is picking those enzymes that do what we want to do. So again, in the case of isobutanol, taking a whole pathway that might take, 20 enzymes starting from a sugar or even back upstream from CO2 to make a chemical with many enzymes that have been adapted from nature to work in a manufacturing setting.
Now, are you aware of, again, I just want to start drilling down into the science and what distinguishes Invizyne. From my understanding is that they've removed the enzyme pathways from outside the cell. So that eliminates the obstacles that you talked about earlier, where you just have limited byproducts, not necessarily high enough yields or titers. Is that very significant? I mean, I think we hear that come from management as an investor, and that's one thing. But to hear from someone that has been involved for 30 plus years in the development, how big of a evolution or revolution is that?
Yeah, it's really a big change. So enzymes typically up until say 10 years ago were used to do one step. So they would take a chemical A and make chemical B, which was great. But oftentimes, for instance, in nature, a pathway in a plant to make a chemical might be 30, 40, 50 enzymes. So the way people would do that was they would try to get an organism, like a yeast or a bacteria, stick those enzymes in the organism and have it do the chemistry in a fermentation setting. So that was kind of the two ends of the spectrum. One enzyme, one reaction, more like chemical manufacturing, simple and easy, clean. And then the other end of the spectrum, fermentation, where you got a long pathway and you've stuck a foreign pathway into that bug and you're trying to convince it to make your chemical instead of, say, for instance, what yeast wants to do is to eat sugar and make ethanol, which is great, but doesn't do much else than that.
Great for wine drinkers and beer drinkers, right?
Yeah, exactly. But not much else. So convincing a yeast, for instance, to make a chemical that you want to use as a fuel has been a problem in the past because, the chemical is often toxic to the organism, so it can't make a lot of it before it starts killing it. And then there's a whole, obviously, the organism is designed or been evolved to grow and make more organism, not to make what you want it to make. So convincing it, and by convincing I mean changing the chemistry inside the bug, is pretty complex because there's thousands of enzymes in that bug, and it's doing lots of things like creating more bugs and growing the cell wall and protecting itself, and all those things make it a super complex way of manufacturing and limited by the toxicity. So take, for example, isobutanol. You can't make over a certain concentration in fermentation because it starts killing the bug. So with the Invizyne approach, taking that just the pathway you want and not the rest of the living organism into the manufacturing setting, you can make 300% higher concentration of isobutanol because there's no toxicity. You're not trying to keep anything alive. Those enzymes aren't living. They're just protein catalysts that are inert. So
right. So we think, and I want you to correct me if I'm wrong with this analogy, because a lot of us are familiar with more immunotherapies and cancer developments where the holy grail there is, like you mentioned before, selectivity, precision, and then low or no toxicity. Is that same, holy grail apply here to synthetic biology, where if you can get those three factors, and that really should produce commercial viable pathways?
Yeah, but you're hitting up against the wall. If you want to use a fermentation, it's good at making things like amino acids or sugars or ethanol, things that don't kill the organism. But there's no way getting around some of these things where, you hit a wall. So in terms of the toxicity, for instance, so getting everything out of the cell and into a stirred tank reactor or a flow column and more manufacturing setting, you don't have to worry about those things because it's not a living organism, so you don't have to accommodate toxicity.
So that's the key advancement that Dr. Bowie and Paul and Tyler have been working on and now bringing here. Are you aware of anyone else I know that there's people that talk about and use the nomenclature cell-free synthetic biology, but are you aware of anyone doing something similar to Invizyne in your experience?
Yeah, there's nobody exactly in the space. There are other people making other chemicals, and so it's kind of a matter of degrees. Like I said, one enzyme, one step, that's the standard. At Codexis, we did, working with Merck, for instance, they developed cascades where it was like 5 steps with 10 enzymes. That's more approaching what Invizyne's doing, but in the case of Codexis and Merck, that was to make an unnatural molecule, which was a drug molecule, and the number of steps was limited to something like 5 or 6. So in the case of Invizyne, we're talking about 15, 20, 30 steps, and we're in taking reactions that are closer to what happens in nature, but pulling them out of the organism and putting it into a cell-free system.
Yeah. You brought up Codexis in your time there. I want to, just kind of tease out from that for people and share with them that you spent, I think it was about 15 years there and ran senior VP of R&D and really helped commercialize their, enzyme engineering platform. So you've not just taken the science, developed it and advanced it, you've seen it move into commercial success and you've been part of that. Can you talk about what it takes to go from the lab to the market, right? What are the challenges? And then how do you see the opportunities for Invizyne in that type of environment?
Yeah. No, so when I started out 2004 at Codexis, that was just the early days of enzyme engineering using these kind of, we were still developing the tools at that point. And one of the projects we worked on was Lipitor. You needed a target that was huge, big in terms of revenue, and it had a big problem, which was the manufacturing, that you could replace it with a cleaner approach. And so Lipitor was the first kind of mega blockbuster. I think it's $14 billion a year revenue at the time we started on it. It was already commercialized. They had a chemical route, and but we could improve that and lower the cost of goods dramatically by using enzymes. And so those kind of programs, the challenges them, it was like, 2 years and 25 scientists to do one enzyme. It was
Wow.
Big spend. Now we're talking about, at Invizyne, it's a few scientists, a few months can do an enzyme. So it's been really a couple of orders of magnitude in reduction in the amount of R&D because of the tools have gotten so much better. We can synthesize DNA cheaply. We can use automation to screen libraries very quickly. We can use machine learning to take the results of our experiments and predict where to go next. And all these things have brought down the cost of R&D and increased the target. So now we can go after things that are, a few dollars a kilogram instead of like Lipitor, which was thousands of dollars per kilogram.
Let's kind of go down into that a little bit more too, because earlier you mentioned that now there's really endless possibilities to explore pathways, what you can produce, what types of molecules, natural and unnatural. How do you prioritize that situation, right? So now you have all these tools, you can do endless things, but then how do you focus what you're doing? What does that decision matrix look like, and how does that apply to Invizyne?
Yeah, so we basically take a product evaluation approach to the product portfolio and say, does it fit with the technology? Are we solving a problem, or is chemistry pretty good at making this molecule? There's some molecules, I don't know, say like ethylene oxide and polyethylene glycol.
What are those? We don't know what those are.
Those are examples of bulk chemicals that are made cheaply using petrochemical derived materials, and they don't need any help from enzymes, as far as I can tell. So that helps define our world that we're going after. Then regulatory, intellectual property, competitive landscape, is the opportunity big enough? Is the delta that we're talking about by bringing in this approach worth the amount of effort that we're spending? So that's how we prioritize our opportunities, to find ones that will give us the biggest bang for the buck.
Have you seen, from your time at Codexis and then moving forward with the development of tools and stuff, what was your experience in terms of selecting a target, finding a partner, and getting to market? What did that time frame look like? Those two steps: target to partner, partner to market. And then how do you think that's changed? Has those cycles been compressed because of the advancements of technology, Invizyne, et cetera?
No, say 15, 20 years ago, you could only go after pharmaceutical molecules that had a really high value and had a big problem with a chemical approach. And they had to have a big market. So those three things narrowed it down dramatically. And so the number of targets that we could go after was quite small. Now it's, as the years progressed, say a decade ago, that opened up because the tools got better. Then you could go after most pharmaceuticals. Now we're at the stage where we're not limited to pharmaceuticals. Now we can go after commodity chemicals and things that are much, much bigger volumes, because we can use a more, now that's, say, an R&D light approach, because we can very rapidly improve these enzymes. We don't need to convince or coax the cell into tolerating this. We're doing it ex vivo or in a cell-free manufacturing approach. So the number of targets has opened up dramatically.
So what about the time to get to partnerships? I mean, what are you seeing? Well, what does it take to get essentially for lack of a better term, the proof of concept to convince a partner you can do what they want you to do and then engage?
Yeah, proof of concept has also decreased dramatically. So it's used to be many scientists, could be 6 months to a year. Now it's 1 or 2 scientists. It could be as short as weeks or a few months to get to that feasibility where you're saying here's an enzyme, it does your reaction, it gives you the product specs that you need, and we can say with relatively good precision how much R&D would be required to take it forward. So we can make a eyes wide open decision: do we go forward with this or not? And so we've had quite a high success rate with that approach because we're getting better and better at doing these programs.
So help me, and I don't know the answer to this. We're familiar mostly with biotech partnerships where there's some, milestone-based payments, certain upfronts. What's in synthetic biology? How do the partnerships typically get structured? And understanding that it may be different in pharma versus a commodities-based market. What were the types of deals that were done at Codexis while you were there that got you to market?
Yeah, I mean, I'll preface it with saying it's really challenging to get paid. So we struggled with business model as the technology changed, right? So, but typically you could sort of capture what you created by selling the enzyme that you've developed to the customer. So you'd together define the approach, say how this enzyme needs to perform and what kind of product specs you need. Once you hit that, and they scale up in our manufacturing, then you could supply the enzyme, and since it's proprietary to, in that case, Codexis, they could charge, a fair amount for the enzyme and try to recover what had been developed. In other cases, people will do it like on, money up front and have no IP ownership, or they might do something on speculation and approach somebody and say, hey, we've done this, do you want to license it? So there's really an array of models. And if you look at the industry, it's really struggled with coming up with the right models. Some people talk about sharing the delta improvement, and
right.
It's challenging, but
so that challenge, the business model has been a challenge to the enzyme engineering / synthetic biology. Scaling has been another challenge that I think has been well vocalized. How do you see Invizyne fitting into that? Where is their approach hitting a sweet spot of the market? What can you say from the science side? Any concerns on their ability to scale relative to other more traditional synbio approaches?
Yeah, no, I mean that's a big part of the cell-free manufacturing is the ability to scale, because you if you're trying to keep an organism alive and say you're making a jet fuel with a yeast, keeping that strain going at large volumes, and they need to be tremendously large, hundreds of thousands of liter volume manufacturing to produce enough of this material, keeping it from getting contaminated while keeping the organism alive is a big challenge, and that's where people have stumbled. Invizyne isn't limited by that. They can get to much higher volumes without, because again, there's no living organism involved, so they don't have to worry about either death of it or contamination with other organisms. And then they can get to much higher titers, so the amount of capex, the facilities can be like tenfold smaller. So the spend and the risk that you're putting up front is 10x less, basically, than a typical fermentation manufacturing.
Yeah. I want to talk and be cognizant of time. We said we'd keep this to about 20, 25 minutes and then open up to Q&A. So I do want to focus really, again, let's go back to the end in mind. You've been in this space arguably for 30, 40 plus years. You have seen big innovations and advancements. When do you think everyone else figures out the potential for cell-free synthetic biology? What's the tipping point from your side of the world where you've had a much longer, broader perspective to see things develop? Is it in the next 6 months, in the next 3 years? I mean, you have a McKinsey Global coming out saying this is the next $1 trillion industry. Where do you see that inflection point happening?
Yeah, no, I think we're in the midst of it. Obviously everything I'm saying here is my opinion, but I think that we are in the midst of it, and Invizyne has the advantage of being, a first mover, and so they've created IP around this approach, and but I think people will see their success, and it'll turn heads, and people will realize that a CapEx light, R&D light approach to developing biomanufacturing processes is pretty attractive. So success will draw in other
Yeah.
programs.
We typically see the market recognition follow. It lags the scientific recognition. What are you seeing from the scientific community, your peers that you've spent your career with? Are they catching on, or do they see, or what's the aha moment for them?
Yeah, no, they're definitely catching on, and I think there'll be a benefit to people that adapt. Tends to be pretty conservative, the industry. So I think Invizyne's approach is going to be working with more innovative companies, and the larger ones will follow later. So there's a lot of inertia around, using petrochemical, obviously, and the facilities are built, and so getting people to shift, I think that will be driven by success, but it's going to take some time. And I think Invizyne will be in a good position to be a first mover to benefit from this approach.
Okay. Listen, I'm going to end with one more question, and we'll open up the question and answer, and it's be a kind of a double-sided one. What's the biggest risk or challenge you see for Invizyne moving forward as they commercialize, and then what's the biggest opportunity? What excites you the most? Put another way.
Yeah, I think it's just the incumbents being resistant to change is always a challenge. On the commercial side, I think I've had cases in my career where I bring a beautiful process to somebody, and convincing them that they want to change with what they're doing is just surprisingly difficult. So I think that's the challenge, is finding those opportunities, the products that need to be made that are also can be done with a partnership with somebody that's willing to take a risk and do something new. That's always the challenge.
Yeah.
Sorry, the second part of your question
was just, what's the biggest opportunity that you see? What excites you the most about being involved with Invizyne, and what's ahead of the company potentially? Again, understanding these are your opinions and forward looking in nature.
Yeah. No, I mean, I'm excited by the sustainable aviation fuel and demonstrating sustainability at that scale, because you've got the benefit of regulatory, especially in Europe, driving adoption of those things. So that's going to give the necessary, I think, protection as the economics grow. Because, when you start out, you're building facilities, putting a lot of capital at risk, you need some basically some support. So I'm excited by picking products that are in that area of sustainability, that fit the technology well, and that have the opportunity to be big volumes and really change the way everybody thinks about making materials.
And do you see those because there's the regulatory pull, or in Europe's case, the mandate is accelerating the process? I mean, are you seeing players more engaged, or innovators more engaged in those markets just because they don't have a choice? Or is there, and again, I'm bringing this up because we've dealt with other emissions control technologies where the industry players could just pay a fine as opposed to complying with the new reg. So is that the case here with SAF in Europe, or are these hard and fast regulations so people understand?
Yeah, no, my understanding is hard and fast, but I'm not in the details of it.
You're a regulatory expert in European regulations too.
No, but the nice thing about it is that with the relatively modest R&D effort that you can go after these big products and go after more smaller volume products as well, to grow the company, to demonstrate the technology, build the infrastructure that's needed to deliver on this. So it's a nice kind of portfolio approach where you've got valuable but more say pharma and nutraceutical food additive type opportunities. And then the big, huge, things like fuel.
Yeah. No, and to that point, I want to just remind people, I think it's in Invizyne's actually the pitch deck that they filed. I want to say it's slide 7 or 8, where they talk about how to de-risk the process and get to market and what the incremental costs are for each step. And if you look at it, if I remember correctly, it's somewhere, those early phases are $50,000 to $100,000. It's hundreds of thousands of dollars to get to a point where it maybe makes sense and a matter of, less 6 months in some cases. And then the spend gets into $1 million or $2 million where you can partner. Is that accurate? Yeah.
Yeah,
Okay.
definitely. Yeah. And obviously much lower barrier to what it had been, say, 10 years ago.
Yeah. Okay. Well, at this point, if you're okay with it, you'll indulge us for a few questions from the audience. Would love to get through that. Again, as a reminder to everyone, the easiest way to ask the question, we're not going to try and put you on screen as well, is just enter them into the Q&A box here in Zoom. We will go through a handful of these here. Just encourage you, please do not ask any questions that would about this specific offering or about forward-looking statements or personal investment advice. We can't provide that. We end up in orange jumpsuits if we do. So Jim and I do not want to do that. So let me start off. We've got a quick question here from David. Setting the green energy label aside, I question the economy of creating jet fuel from sugarcane or corn. Is the production supply sufficient and sustainable? Is it cost competitive with fossil fuels? The biomedical applications seem to make more sense. So there's a lot there. If you need me to kind of unpack it piece by piece, I think first is, does Invizyne, if we get into the SAF, is it competing with the food supply? And if so, how do you balance that?
Yeah, no, obviously there's been a huge effort in this country and worldwide in biofuels and people looking at supply chain. And I'm not an expert in that, but my understanding is, for instance, if you look at Brazil, where you've got sugarcane to make sugar and ethanol, you also have bagasse, which is waste, agricultural waste, the cellulosic that you can drive sugars from them. So there's a way to sort of co-produce and take value from what would be basically a waste value.
So you can use the corn husks and that waste to drive these enzymatic reactions.
Yeah.
Okay. So I think that answers that question. The next one is, Jim, what is your opinion of the status of the synbio sector from the viewpoint of the public companies that are out there? Do you feel that we're near a more positive time for the group based on the technologies, or is this sector going to continue to struggle because of the technological shortcomings of those companies that are already in the market?
Yeah, no, I think the technology is moving forward really nicely, and I think companies that are able to capitalize that will benefit. I don't want to comment on the abilities of specific companies, but just like any technology, like we had the booms and busts and boom again of the internet and .com and now AI. It's really akin to that there'll be people that fall out and people that succeed, and but it's going to be, in my opinion, trillions of dollars produced by biological means. And I think
so without naming names, let me try something here, and you can refrain from answering if you choose. Were there any other companies that are public now in the synbio sector that you were approached to join as a board or scientific advisory member that you declined?
Yes.
Okay, fair. The shortest answer of the afternoon.
Might also say, just like .com and all that, you've seen, like, people thought, say 10 years ago, it would be, synbio, and even where the term came from was we could just treat it like coding and DNA. You could just stick it in a bug and it would just do whatever you want, which was obviously a huge oversimplification and turned out to be wrong. The bugs don't do whatever you want; they do whatever they want. So, just like in the internet and computers and AI, our field has also had these booms and busts and booms again. But I think it's all part of becoming a mature field.
Yeah. I want to end with just kind of, I would say, this is my personal opinion, when looking at investments, the toughest thing to control for is management, the team, humans, right? You just don't know So I want to ask you, given your experience on the science side, but then also with commercial applications with Codexis, what's your evaluation of the team, the executive team that's there, from Michael Heltzen and Zach, and then the science team with Tyler, Paul, and Dr. Bowie? And obviously, I want to say I have tremendous respect for all of them, so I know I'm leaving out some of the team members. So I want to be all inclusive. What are your impressions, given your experience and working with other organizations?
Yeah, no, I mean, that's how I got involved, actually, just from Jim Bowie's publications and meeting Tyler and Paul at conferences and learning about what they're doing. And then I saw an opportunity with some of their work that they published on cannabinoids to apply some technology that I knew about, which was a cofactor recycling. And that's how we kind of collaborated at a scientific level, say 5 years ago. So I've been connected to them since then, and really impressed with their ability, their creativity, diligence, and really cutting edge science. And now that the executive team has been brought in, I'm really impressed with their acumen and their expertise in the field. So as you kind of hinted at, the team is everything, and you can see companies with great technology and they have some political problems inside, and they don't make it, right. So it's huge, and I'm really enjoy working with this team and really impressed with what they've done.
Well, I enjoyed the conversation today. I really appreciate the time you've taken. I'll give you an opportunity if there's any final things that you'd like to say, but by no means no pressure.
Yeah, no, just super grateful for being a part of this field and to be a part of the Invizyne offering. So it's
well, we're thankful to have someone with your science stature and then commercial stature join on the team as well. I could say that from my personal standpoint on behalf of the MDB Capital team. So I would like to also thank everyone for participating today. If you have any questions whatsoever as a follow-up, you can always email us at community@mdb.com. As we mentioned at the outset, we have recorded this and we'll be posting the recording to the website at Invizyne, as well as relevant social media platforms here shortly. So again, thank you, Jim. Thank you, everyone else, for paying attention today, and look forward to the developments that are ahead for Invizyne and the field of synthetic